So today I'm thrilled to join you and to participate in your One Health Hackathon, at least by providing some background. I'll hopefully leave some time at the end so we can discuss with questions. I know that you've been already started and you've been working on your hacks, but I just wanted to bring back in the definition of One Health being a collaborative, multi-sectoral, and transdisciplinary approach, working at the goal of achieving optimal health outcomes and recognizing the interconnection between people, animal, plants, and their shared environment. So there was a shift fairly recently, just in the last couple of years, where we pulled plants out of the environmental aspect. And I think that's a really healthy change, because we used to put plants sort of absorbed into the environmental scope. And if I'm really honest about working in One Health, I think that we have not been able to fully embrace and bring in the environmental community and the plant health community. And of course, plants are so critical to feeding the planet that I think they do deserve their own circle. But we still need to work harder to bring in those environmental aspects, but also the environmental experts, as well as the plant experts, into the One Health working arena. So when I think about One Health, I use that definition. I'm really happy with that definition for a couple of reasons. One is that I think of One Health as being an approach to problem solving, not a discipline. And so if we think about it as an approach, then we can recreate it every time we're dealing with a problem. We can bring in all of the important disciplines and be open to those disciplines or identifying those disciplines as we go, because sometimes you start and you think you've pulled everyone in, and then some revelation happens and actually you're missing some expertise. And so be open to bringing people in as you go. And I also, I'm going to talk to you a lot about zoonotic disease emergence today. So those diseases, those pathogens that can shift from species to species and then make people sick. So I'm going to talk mostly about that, but I wanted to just list some problems that we've used the One Health approach to help address here on the right side of the screen. And I really wanted to highlight for you that the drivers, or the things that sort of cause these problems to happen, are often the same for more than one of the problems. So if we can go upstream and really work on those drivers like land use change, climate change and the climate crisis, economic development, poverty, social inequities and justice, environmental justice, if we can look at those drivers and start to actually address and solve some of those issues upstream, then we're going to have many fewer consequences downstream, meaning the problems. We'll have less problems if we can work on the drivers upstream. And as we're using the One Health approach, we always need to keep the influences that I have here on the lower left in mind. So you have to think about culture, economics, policy, behavior, education, all the underlying pieces that form the society and the communities that you'll be working with. Now, the One Health interface, again, I'm speaking about emerging infectious diseases because that's what I do, but I don't want you to think that the One Health approach is only useful for emerging infectious diseases. It just happens to be the example of where it's really obvious that we need this multidisciplinary collaborative approach. But there are so many more problems where we can use the One Health approach. And I know that your other speakers and you, as you're doing your hacks, are really thinking about those. But it is the case that the majority of zoonoses, again, those diseases primarily that we share with animals, have wildlife origins. And the majority of emerging infectious diseases in people are zoonoses. So both that we share with animals is important, and then the things that we share that are new are from wildlife primarily. And that's really because we're changing the way that we use the planet. We're changing the way that we use land. Our population is growing, and that is causing us to come in contact in a more intimate way with many more species that might have lived in pristine areas and things. So it's not surprising that diseases that we are getting because we are naive to those diseases are coming from animals that we probably didn't interact with as much or as intimately as we are now, as we're sort of spreading across the planet. So all of those things cause the ability for us to be exposed more often. Again, our behavior, no virus or wildlife is jumping out to infect us, right? It is our behavior and how we are using the planet that is causing us to be at increased exposure risk. So then we have an enhanced flow of pathogens, health risks, not only to the humans, but also to the other species. And that can cause really economic consequences and the way that we live our lives consequences, as we all know, because we're doing this remotely. And then that is just finishing the circle and starting it again, because that means then we change our behavior again, use the land differently, maybe move, maybe migrate, and it just keeps the whole cycle going. So we know we're vulnerable. I hope that that the majority of you here are well and healthy and haven't suffered devastating family losses or economic losses, but we know that is happening to many, many people, millions actually, around the planet are so suffering so much. And every single one of us has been impacted by changes to lifestyle and worries. And those things are just devastating. And we're going to feel that for decades to come. But it isn't new. We are vulnerable, and we were seeing, even in the past decade, on the order of 3 new emerging infectious diseases in people every year. And again, the majority of those being zoonotic. And now the projections are looking like we're recognizing at least 5 emerging infectious diseases of people each year. And I think that's just going to continue to go up. A little bit of that is because we have better detection, but the majority, even if we control for that better detection, better surveillance, better diagnostic tests, is really, it still, if we control for all of those factors, these things are on the rise. The emerging infections are on the rise. And again, I think that goes back to what I was showing you on the earlier slide. So obviously, we would hope never to be exposed and become infected and cause an epidemic and then a pandemic to something as dangerous as COVID-19 again. But if we don't change the way we think about these things, use the One Health approach, we are just repeating history. We're just sitting here waiting for the next thing to hit us and then trying to use all of our resources to attack it. Now, we can't eliminate every spillover, but we can identify the health risks early and try to get ahead of them so we reduce our risk. We also can be ready at the source so that when there is a spillover event, we can try to control it more effectively at the source before it gets out of control. So I just want to give you some perspective here. When I started working in this realm about 20 years ago, there were about 250 zoonotic viruses known to make people sick. Now there's about 260 viruses known to make people sick. When we go through the full characterization to officially name them and understand the viruses. But as we've been working on a project that I'll tell you more about as I go, we have identified that there are probably 500,000 viruses out there still to be discovered that can infect us. And of course, we want to know as much about them as we know about the ones that are known, as we know about the bacterial communities and how they're changing. But we haven't been doing that work. Only a few projects around the world have been really working hard to find those viruses. But it's completely within our control to do that, to find them, understand how they're interacting with their hosts, how we put ourselves at risk for exposure, and reduce that risk. We just have to do the work. So here's an epidemic curve, and I am an epidemiologist. Thank you again for that nice introduction. And what I'd like to just show you is for those of you who are more visual and think about things in graphs and charts, I just want you to think a little bit about how this might be occurring. So if we have a standard epidemic curve, that's the red one. When we think about human infections and we count the number of cases over days and weeks and months and now sadly years, we can have that epidemic curve. And that's the place we don't want to be. We don't want to be in that epidemic curve at all if we can help it. And if we are finding ourselves inside that epidemic curve where people are getting sick, we want to be able to have measures to jump in and sort of control that epidemic. So I believe we need to go upstream into the green area. That's what we call the zoonotic pool, where viruses are sort of circulating in their own hosts. And if they evolved in those hosts, they generally don't make those hosts sick unless they're debilitated, old, have some other stressor. But they do have a normal cycle. They're living symbiotically with their microbiome and virome. And so those viruses are available to spill over. So when there's close connections between domestic animals or people and wildlife, we have the opportunity for exposure. And that spillover then can happen into domestic animals, like the story with MERS and the camels, and then it can cook along, maybe change, maybe become more virulent or more transmissible to people, or maybe not, maybe just stay the same. And then though, it can spill over into people, or it can spill over from the zoonotic pool, the green area, directly into people. And what we really want to do is get better at understanding what's in that zoonotic pool so we can forecast and be ready for anything that might spill over. Then we also need the laboratory systems for early detection. Again, all of that should be and could be prepared before the first case of human illness, and preferably before any livestock or other domestic animals get sick. And if we have that, we're able to come in with control early. So you see the orange circle. If we do that, we move the domestic animal blue curve and the human red curve over to the right and slow it down and potentially can stop it. But when we're not ready, knowing what to expect and being able to detect it, we have the curve that you saw before. So I'm going to tell you, a little bit more about my experience working with amazing people in 35 countries around the world, trying to do just pilot what I just was indicating, and that is to actually find the viruses ahead of time, help countries. I wish our own was ready as some of these countries where we work, to really put the systems in place and have the laboratory capacity, the trained biosafety, biosecurity people to be able to do this kind of surveillance. And preferably we do that by getting ready early. And here's how we did it. We worked at that zoonotic pool, a little bit different graphic here for you to see, but we worked in this viral pool to identify in healthy animals all the viruses they might have, and really get to that early recognition. And then we also looked in the domestic animals and the people that were interacting with those wild animals. So we could try to see, are the people harboring the same viruses? Are they sharing the viruses that we're seeing in the animals? And we wanted to look at both people who might be sick. So they're at the clinics, they're at the hospitals in the areas where people are interacting with wildlife, and those people that have high occupational risk. For example, they might be bat guano harvesters. And we wanted to see if they had higher proportions of these viruses. So that's where we started in our philosophy. And here's what we did. We were out, and I'll be honest, we swabbed a lot of butts. So we're out there looking in camels and chickens and monkeys and pigs and bats, and working in markets where people are buying these animals for food. And we're looking not just at the animals you can see here, but also the animals that you can't see, like the bats and the rodents and the shrews and other creatures that are much more cryptic, but are in all of these ecosystems that you're seeing here. So just wanted to sort of highlight that this work isn't very glamorous, and you have to take high biosecurity measures, but you have to work with the communities. And frankly, the communities were so wonderful. Where you see these chicken and pigs in the lower left, those folks, that is just outside the front door of families' homes. And in fact, sometimes when the animals are sick, they bring them into their homes. And so we worked in these communities to say, do you want to know what we're finding in the animals? Would you be willing to share with us information on wildlife in your area and how much you're interacting with wildlife, how much you see the interactions between non-domestic species and your domestic species, how many, rodents and things you get in your houses. And then, oh, by the way, would you also provide some samples to us biologically? And people were so willing to do it. They went in their back room, they swabbed themselves through the nose, everything else, and brought us those samples back. So it was a really great community effort. As long as we were partners with the community, hearing their concerns and then bringing their results back to them. To do all of this, we had to really train a disease surveillance workforce. So we had to have biosafe individuals wearing full PPE and sometimes testing people's food in full PPE. And so there had to be a lot of, again, pre-work with the communities so that they would understand why we were being so safe, why we wanted to look at their food, and help them understand what was there and make their choices to be safe as well. And then when we were in the field and sampling, we could get that sample once we had all that training done and safety protocols followed, but we had to get into the laboratory and find a way in a cost-effective manner to actually test the samples for things that no one knows whether they exist yet or not. So that was a very difficult challenge to overcome, both getting the equipment and training into the laboratories, but also working with those folks in the labs to figure out what is the best way, most efficiently and effectively and inexpensively to be able to test the samples for the world's most dangerous pathogens that have not yet been identified. And so what were we able to do? Well, over the lifetime of the project, and I'm reporting on about 11 years of work here, we trained almost 7,000 people in the One Health approach. Also, obviously, to be safe, to transfer samples safely, to handle them safely, but then to detect the viruses, identify them, and report them to their country governments and share them with the general public for better global security. In doing that, we were able to work with 60 laboratories and get them up and doing viral discovery. And they had this wealth of data and samples coming from the field as we were able to sample about 164,000 individuals, most of those wildlife, many though humans, and some livestock. So very specifically, We were looking at 164,000 individuals, but we had 9 to 12 samples from each of those individuals. So this was a huge data project. In the end, we discovered about 1,000 new viruses. And by discovering those viruses and sampling the same species over and over, we could come up with that estimate of about how many viruses were left to be discovered. And that's where that 500,000 number that I shared earlier comes from. We also were able to detect more than 200 known viruses, but those viruses we were finding in new areas and in new hosts, which helps immensely with understanding risk reduction. So we can actually look at, for example, if we find Ebola virus in a new host or we find it in a new location, that is useful for where we should be working on risk communication, risk reduction, reducing exposure. One example is we did find a new Ebola virus, so not the ones that are known to be causing these devastating outbreaks, but another whole species of Ebola virus. By working with the communities who were devastated by the West African Ebola disease epidemic, we were able to be in their homes and look for that disease and the hosts of those diseases while we were also looking for other new viruses, and we found one. And that was a very interesting challenge because we needed to understand in what hosts are these is this new virus found, and we found it in more than one species of host, and how are people interacting with that. And then if you look on the right here, you can see how the home range map of the host, the bats that we found this new Ebola virus, how it looked across Africa. And so those home ranges of those hosts that we found should give us the perspective of where are we going to find that virus. So we found it here first in Sierra Leone, but as soon as we started communicating and sharing that information, another team found it in Guinea. And then our team also found it in Guinea. Then another team found it in Kenya. And then we found it serologically with evidence of exposure in people in DRC. So once you find these new viruses and they're of interest, it's much easier to improve the surveillance, and teams that are not necessarily your team can start working on some of these. Now, we did a lot of outbreak support. We responded at the request of country governments to more than 50 outbreaks. Most of those viral hemorrhagic fevers, like is caused by Ebola Zaire and Ebola Sudan, but also just straight up unknown diseases, and then diseases that were unknown when we started responding and became known. Some of those things like avian influenza, monkeypox, other things that you would know about. Now, we didn't identify SARS coronavirus 2 before the terrible pandemic started. We were not working in that part of China, but our work did help to identify and put some scope around what hosts might we be interested in looking at and exploring so that people don't continue to get infected. What interfaces are most highly at risk for causing or allowing exposure and spillover. So we were able to use our data, for example, on the wildlife trade and mixed animal markets to help understand where risks might be, and past serologic evidence of people who've been working in those markets and how often they appeared to be exposed to things like coronaviruses. And our teams all over the world, but especially around China and in Africa, jumped in straight away. So those 60 laboratories I mentioned and all the teams in the countries, even though actually our project had finished up in the field and we were no longer working in those countries, our teams were able to jump in and working for their own governments or their own universities. They were the front line for COVID prevention in the case of Africa, or getting ready, and identification of the first introduced cases into their countries. So for example, in places like Thailand and Nepal, the teams we had been working with identified the first introduced cases of COVID-19 into their countries at their borders before we even had a diagnostic test for COVID-19 that came from the WHO information. So they could use the PREDICT protocols and find that virus and say, yes, this is SARS-CoV-2. It didn't even have a name yet, but it's coming into our country. We need to get ready. We need to put in place our public health system right now. We put these people, get them into quarantine, get them well, and help to start doing specific follows of their contacts to make sure that they're not sick and that they're being careful and sheltering so they don't expose others. In Africa, in places like Kenya and Rwanda, our team members were some of the first people, maybe the only people, to ever have done a scientific investigation and written a manuscript about coronaviruses. And so they were obviously the frontline resource for their country governments. In Tanzania, our team member was put on the national task force, and in many other countries, there are similar examples. So I think we were really helpful, and all these icons you can see here are showing you what activities our teams were doing for COVID-19, even though the project had wrapped up. And we were able to reactivate the U.S. Agency for International Development, gave us some funding to go back to our freezer archives where we had sampled potential hosts, and also help those teams do additional testing. So we reactivated the network with some support from the U.S. government. The other thing is all of our data had been generating a lot of information on SARS-like coronaviruses that we were finding. And for example, we were able to identify that horseshoe bats, as are shown in this picture, are a host of SARS-like coronaviruses, and that they use the ACE2 receptor that people are talking about so much now, just like people do. So they didn't necessarily need a different intermediate host for the virus to go into and evolve and change and then be able to spill over in people. It could come directly from the bats to people. So that science was informing much of what was going on. In addition, these are all the coronaviruses that we detected during the project. We detected 177 unique types of coronaviruses. We identified them thousands of times, and 160 of those 177 were brand new novel coronaviruses. So that information allowed us to start to break down, where are we seeing these different kinds of coronaviruses and in which hosts? And the Sarbeco coronaviruses, the SARS-related coronaviruses, the 2B, also known as 2B coronaviruses, we found dramatically associated with rhinophyllid and hippocyterid bats as the hosts when we were finding them. So you can see here, more than 75% were in these taxonomic groups of bats. And so that, again, helped the team say, here's where we can expect to see issues if we're not starting to prepare. And for SARS coronavirus 2, we overlaid and showed the distribution of those host species and saw that the risk is not isolated to just the Wuhan area of China. It's actually all of central to southern China, across in South Asia to Nepal, and then throughout Southeast Asia, just really heightening where surveillance should be done for these viruses. We also could look at those interfaces and how risk is amplified across some of these interfaces. So for example, we were looking at rodents for coronaviruses and other viruses in Vietnam. And what we found was when the rodents were in the wild, they had about 20%, just over 20% prevalence of coronaviruses. But then when you put them into the value chain, meaning they were going into farms to be reproduced, bred for the food market, or taken straight into the markets and the market chains, but then they were sort of cramped into small spaces, so they're able to expose each other, and then mixed with other species, which means they can expose the other species, including humans, but also potentially allow for mixing of viruses that those other species have with the ones they had. In that large market, we saw that the prevalence, or the percent positive, went up to over 30%. And most worrisome, we saw that by the time they had been in that value chain for some time, long enough to get into become food, so at the restaurant interface where people were butchering them, cooking them, or preparing them in some way to serve, we found that more than half of the rodents were infected with coronaviruses. Again, identifying where these risks really needed to be reduced. Now, I want to make sure that as we're talking about the wildlife hosts, and especially for coronaviruses, we're talking about bats, I want everyone to remember that our job is also to help people understand how ecosystems function and why we need bats. So even though bats do seem to be an ancestral host for many coronaviruses, we need them. We need to protect our biodiversity and our ecosystems, and bats provide key ecosystem services. They control pests, they help to pollinate, and so that helps us to have better, healthier crops and food supply. The fruit bats are seed dispersers. So as we fragment landscapes and break them up with roads and cities, and we have a pristine landscape over here, maybe with endangered trees, but there's not enough room for those trees to live and exist and not go extinct without other landscapes, but they can't communicate across long distances with their gametes, right? To be able to continue the species. The bats provide that link because they happily fly right over us to the other pristine fragment and share the seeds, so that we have better, more healthy, biodiverse planet. The other thing is, if you try to kill bats, reduce them by culling, those efforts have been generally unsuccessful. They come back and they start to increase their reproduction. And when they increase their reproduction in response to culling or being killed, what we found is that that's when they shed more virus. So we were able to document that bats really shed the most virus while they're weaning their pups. And so that helps the virus be able to get into a new whole age cohort, but it also provides us information about when is most risky for people to get exposed and how we might reduce that risk, especially at critical times when the populations are shedding viruses. So we developed a community outreach tool called Living Safely with Bats that talked about these benefits and risks and how people could be safer. We did this in the Asian context and also a version in the African context. And then it was translated by many into many languages, more than 12 that I know of, and then delivered in even more. So this curated kind of outreach tool is a book that we can print and people can take with them, but we prefer to do it as a poster kind of session at a community meeting. And there's a narration, there's a script that the people delivering it can translate into the local language as they're talking to the communities. Now, the next phase of where we need to go with this, in my opinion, is that we need to think about doing things differently. So I already introduced that to some extent by saying that we really need to get ahead of those curves, right? To the left side of those epidemic curves. But to do that really well, that means we need to discover all of this virus. So we need countries all over the world to be participating, looking in their own country. If they have partner countries that maybe can't afford to do it on their own, they need to help those countries, as the U.S. does through the U.S. Agency for International Development that sponsored this work. And so I'm participating in an initiative that we're calling the Global Virome Project. And that's trying to get this partnership of countries all over the world looking for these viruses. We had started before the pandemic. We had to kind of shut down as we didn't want people that might have COVID-19 providing back into wildlife populations that weren't infected the virus, right, SARS-CoV-2. So that kind of shut down a lot of our work for now. But in addition to that, we think we're ready for the teams to jump back in. And now, because of the terrible pandemic, many more will be interested. So we are capable. We know with PREDICT, we were able to show that we can test species, we can discover the virus, but now we need to go beyond what we did, which was looking primarily in rodents, bats, and non-human primates, out to all the mammalian and waterbird species that interact with people, and get them tested, discover those viruses, and use those viruses to inform strategies to reduce risk by stopping spillover, changing our risky behavior, but also providing watch lists as we're finding those viruses of which ones might be worthy of further investigation, maybe active surveillance, maybe even thinking about how we can improve the diagnostic pipeline and the vaccine pipeline. And so through this global network, then that data can be used to really help us have better global health security. And one of the tools that our team developed, because we were discovering so much virus and we didn't want to just say, well, here you go, you have these viruses. We wanted to help put some interpretation around that, help the public health officials in their countries decide where they might strategically increase surveillance or stage equipment for response. So we had to take all of the information that was out there in the scientific literature and combine that with expert opinion, because there wasn't necessarily a great risk ranking platform. Here's what makes viruses risky. So we had to go out there and ask the experts all over the world about this, and then use that to combine with other global data sets about the locations where we were finding the viruses and the hosts where we were finding the viruses, to really get a handle or start the conversation so that we could get a handle on which viruses are risky. So we actually looked at about 42 different risk factors that are here on the right. And I took those to the international community, the experts, and asked them, which ones of these do you think contribute most to spillover risk, and which ones are less important? And we ended up using 31 of these risk factors that were ranked as moderate or high risk to look at all the viruses that have been found in wildlife, in taxonomic families that have been known to cause human disease, as well as all the viruses that we were finding in PREDICT. And with that, we wrote a paper, yes, but we also made this tool that you can visit. If you go to www.spillover.global, you can see the data, you can play with the data. So you can look at the risk ranking or watch list. And for example, you can make your own. This is the coronavirus watch list. So we could just, if you're interested in coronaviruses, go in and say, based on those 31 risk factors, which ones are most worthy of further surveillance and concern, and are they ones that have caused disease in people before? Or you could, if you're specifically interested in a specific virus like SARS coronavirus 2, you can dive down in and look at every one of those 31 risk factors and see how much each of those risk factors contributed to the ranking of the one virus you're interested in. And I've shown you here SARS-CoV-2. For public health planners and officials in countries, you could make a watch list for your country. So this is a watch list for Uganda, for example, that includes the known and the new viruses that are harbored in wildlife that might be available to spill over to people. So you can see Marburg virus is at the top of the Uganda list. It's caused terrible disease in Uganda. So that's not surprising. But then you can drop down and start to see some of these bat viruses that maybe are not known to cause disease in people yet, but are there in Uganda and should be considered when and if there are cases of strange disease. In addition, the scientific community can add their own viruses. So we have, with just answering about 10 questions, we can pull from those public data sets all of the risk information for the 31 risk factors and provide the investigators who find the viruses with how their virus ranks in what we already have on the website. And then if they're willing, they can submit it into the public domain and share it with the world through this interface. Okay, so that's an example of what you can do with the One Health approach if you're jumping in and really bringing the different disciplines together. But it doesn't start there. It starts where you are. It starts with students. It has to start early. And so we're very committed to helping to just make a strong One Health pipeline and train the future leaders, the next generation of One Health workers. And I just wanted to share a couple of examples here with you. On the right, the people in scrubs, those are our veterinary students that actually work in clinics for underserved communities. And on the left, that's our network called the One Health Workforce Next Generation Network, another project sponsored by USAID, where we're working with all sorts of disciplines throughout Africa and Southeast Asia, and they have been building their university networks to really infect all of the disciplines at the undergraduate level with the One Health approach. In addition, and if you're interested a little bit further on in your career, we have a field course called RX One Health, where we bring professionals, sort of early career professionals, graduate students, professional students, and people who are already working but fairly early in their career. We bring them together, build a cadre from all over the world so that they're connected with maybe an MD who came from Los Angeles, is connected with a environmental scientist from Uganda and connected with a public health professional in Vietnam. And they are staying together as a cohort and leaning on each other when they are going through their careers and finding other important things, because they all had this intensive life experience for a few weeks of living and working together and learning about One Health. So I hope that you will join me and take this perspective that we need to tread more gently on the earth and work together to really have a better future for the planet, including prevention of pandemics, which is within our reach. And what you can do, obviously you're really motivated and you're doing the One Health Hackathon, but you can also listen, learn, reflect, and act. Please recognize and address inequities. Think about Black Lives Matter movement, thinking about social and environmental justice in everything you do. Bring this transdisciplinary approach and professional respect across different areas of expertise. Bring that with you throughout your career, and you will have so much more of an impact. And then make sure you're always thinking and working with the communities, not as some group that you take information from, but as partners who inform your study design and allow you to actually have that living laboratory and your partners in communication when you find something, so that they can take their health into their own hands. And so with that, I'd just like to end and open it up to questions.